In our previous experiment, we used chromatography to separate a mixture of dyes into their individual components. Chromatography works by distributing substances between a stationary phase, like silica gel, and a mobile phase, which is the solvent. Dyes that interact more strongly with the stationary phase move slower, while others move faster, allowing us to separate them as they travel through the column. After isolating the dyes, we prepared them for UV-Vis analysis by creating diluted solutions. We transferred each collected dye into a cuvette and diluted it with deionized water. This dilution step is important because overly concentrated solutions can give inaccurate absorbance readings. These prepared samples are then ready to be placed into the spectrophotometer, where we will measure their absorbance and analyze their spectra. Next, we prepare the spectrophotometer by setting the wavelength scan range from 800 to 200 nanometers so we can capture the full UV visible spectrum of our dyes. Before taking any measurements, we need to zero the spectrophotometer using a blank. We previously filled a clean cuvette with deionized water, which served as our blank solution. Before placing it into the instrument, we made sure to clean the two clear sides of the cuvette to remove any fingerprints or residue that could affect the final reading. Once the cuvette is clean, we insert it into the spectrophotometer and zero the machine. This step is important because it accounts for any absorbance from the solvent, cuvette, or instrument, ensuring that our final readings only reflect the absorbance of the dye itself. After calibrating the spectrophotometer, we begin analyzing our samples by placing each cuvette into the instrument one at a time. For each dye in the middle fraction, we run a full scan from 800 to 200 nanometers to measure how much light is absorbed at each wavelength. As the scan runs, the spectrophotometer generates a spectrum that shows peaks at specific wavelengths, which represent the wavelengths where each dye absorbs the most light, also known as the lambda max. We record the wavelengths of these peaks for both of our separated dyes as well as for the middle fraction. The middle fraction is especially important because it may contain a mixture of both dyes, so we expect to see multiple peaks in its spectrum. By comparing the wavelengths we obtain to known reference spectra, we can identify each dye and determine how pure our samples are. This process is important because it allows us to use absorbance patterns, not just color, to accurately identify substances. It also helps confirm whether our chromatography separation was successful or if there was still overlap between the dyes. Beer-Lambert's law describes the relationship between absorbance and concentration and is written as A equals epsilon times L times C. In this equation, A represents absorbance, which is measured directly by the spectrophotometer. Epsilon is the molar absorptivity, which is a constant that depends on the specific substance and the wavelength of light being used. The variable L represents the path length, or the distance the light travels through the sample, which is typically 1 centimeter for a standard cuvette. Lastly, C represents the concentration of the solution. The values for absorbance are obtained from the spectrophotometer, while the path length is determined by the cuvette being used. The molar absorptivity is usually known or determined from a calibration curve. According to the Beer-Lambert law, absorbance is directly proportional to concentration, meaning that as the concentration of a solution increases, the absorbance also increases. This relationship allows us to use absorbance data to determine the concentration of an unknown sample. Spectrophotometer data can both be qualitative and quantitative. Qualitative data helps us identify a substance based on its absorbance pattern across different wavelengths, while quantitative data gives us exact absorbance values that can be used to calculate concentration. We scan across all wavelengths to see the full absorbance spectrum and to locate the lambda max, which is the wavelength where a substance absorbs the most light. This is important because using the lambda max gives us the most accurate and sensitive measurements. Another reason we scan across all wavelengths is to check for multiple peaks, which can indicate the presence of more than one substance, like in a mixture. After collecting our absorbance data from the spectrophotometer, we use both the absorbance values and the spectra to identify our unknown dyes. We looked at the wavelengths where each sample had its highest absorbance, or lambda max, and compared those values to known reference spectra. We also considered the absorbance values we gathered for our two dyes in the middle fraction, since differences in these values helped us recognize if a sample was more concentrated or possibly a mixture. By matching the peak wavelengths and the overall shape of the spectra, we were able to determine which dyes were present. This works because each dye has a unique absorbance pattern, almost like a fingerprint.